microscopy, and the name itself, it is self-explanatory, micron. So it is a small, and also the scopes to look, to look. So in that case, to look the small objects without the aid of human eye or a naked eye, how we can see the specimens. Okay, so microscopy is the field of using microscope to view the objects and the areas of the objects that cannot be seen by the naked eye. Okay, so if I say that some Vijay is present here, so immediately you are able to stand up. I am able to see you. That is, with my naked eye, I am able to see you. So if it is a very small bacteria, we are not able to see that. In that case, the external aid is necessary. So for that, we are using the microscope. So the study of microscopes are called as microscopy. So it is a science of investigating small objects using the microscopes. So it is a very good field, and if you know nook and corner of the microscopy, so it is very easy to even become an entrepreneur. Okay, so there are a lot of biological specimens have to be dissected and find out what are all the variations in anatomy, or what are the structural modifications occurs in that, what kind of organisms are present in the environment. So everything, it can be dissected out with the help of different kind of microscopes. So in that, they divide this microscopy into 4 branches. That is, to observe the object, if you are using the light as a source, then we are calling it as optical microscopy. Then electrons are embedded to focus a particular object, that is the electron microscopy. Or you are using a probe. Probe means certain sequence or certain material is used attached with a particular tag, and if that particular tag is present, then we are saying that this object is present. So based on that, you are able to scan the probe. So it is scanning probe microscopy. Then the last advance is the atomic force microscopy. So we are seeing at the molecule level, at the atom level, how the electrons, protons and the neutrons are constituted in that specimen. So that can be analyzed with the help of this atomic force microscopy. Okay, so we will see one by one in this class. Okay, and in general, if you are measuring a particular object, the metric units are used. So in that, you start with a meter. Okay, so in that, so we call it as 1/10 of a meter is decimeter, but generally we won't use that as decimeter in a common measurements. Immediately after a meter, we are going for centimeter. Okay, 1/100th of a meter is centimeter. Okay, then after that, 1/1000th of a meter is millimeter. Then 1 in 1 lakh, sorry, 1 in 1 million, it is a micrometer. The micron is the unit used. Okay, micrometer, 1 followed by 6 zeros. So 1 in 1 million. So if it is a million, there should be an equivalent, another one, 1 in 1 trillion. Okay, so in that case we call it as nano. Okay, so 10 to the power of minus 9, 10 to the power of minus 6 is micron. Okay, so 10 to the power of minus 3 is millimeter. Okay, so like that we are using the metric units to, based on the size of the objects, whether we are observing at the centimeter scale or measuring at the centimeter scale, at the millimeter scale or micrometer scale or nanometer scales. So measuring at the centimeter scale, you can take a scale, measure the viewable objects with our eyes. Okay. So if it is not possible, then we have to keep it under a microscope and then we are measuring them. Okay. So that the micrometers are useful. Okay. So up to, this is a 15.3 micron. So micrometers. So in that case, the length of a bacteria or a width of the bacteria or length of the flagellum in a bacteria, so everything can be measured in terms of micrometer. Okay, and if you go little bit advanced in the images, if you want to see a very fine structure, so you can measure with the help of nanometer also. Okay, even the advances came it for picometer also. Okay, another 3 zeros you can add, but mainly up to nanometer you can use this. So what are the biological organisms? What are the examples for that? For example, if it is a tapeworm or any worm, you measure, you can measure in meter. Then the another 1 centimeter, you can measure a diameter of a mushroom cap. Anything you can write an example, centimeter with a centimeter scale. Then millimeter, the diameter of a bacterial colony. You are taking a Petri plate, you are inoculating with a particular medium, and in that you are growing a bacteria. The bacterial colony is growing. You want to measure, and that is in case of millimeter. Okay. Then if it is a micrometer, so the diameter of the red blood cells. Okay. So that comes under the micrometer. That is red blood cells. You have to observe under a microscope and then see that. Observe under the electron microscope and see the virus particles. So how much nanometer they are measuring. Okay. So that comes for the nanometer. Okay. So the first microscope was invented by a father and a son. Okay. So have you discussed anything science with your father? Anyone in this class? Science with your father? Anyone? Raise your hand. Good. Then something, okay. So the son is playing with some material and the father is observing it very carefully, and then he found that something different in that, and they are able to see that there is some magnification in the view of that object. Then they thought that, so this can be used for studying the various objects. Okay. So like that, the Hansen, Janssen, they are the father and son, and they are from Netherlands. They invented the first microscope. Even though it was not much publicized, immediately if you say the microscope means Antony van Leeuwenhoek is the answer for everything, father of microscopy, like that. But before that, even with the in 1580 to 1638, these two members in their home, they are able to play with a instrument and to magnify the object, but they are not published. So if it is not shown to others, it is not possible to record it. Then next to that, the Antony van Leeuwenhoek is the inventor of the microscope first. So he made a simple microscope that could magnify about 275x magnification, and he published. See that? So once you record the observation and record it in a scientific journals, then only your discovery or invention or innovation, everything will be taken care of. So like that, in 1683, he published that paper, and he told that with a simple microscope, it is possible to see the objects to the magnification of 200x. Okay. So this is the claim of Antony van Leeuwenhoek. So he is the inventor of the microscope. Okay. So what is the simple microscope? So one lens you can easily observe. Okay, if you go to a palmist, okay, reading your various rays, okay, then immediately what he will say, with the help of the magnification, 10x magnification, he is able to see your palm and say that you will come very good in your life, like that, isn't it? So just if you are saying that it is a 10x magnification lens, or sometimes they will use 5x also, but the same 10x lens are used in a small instrument. Okay, in that a small lens is put it at the top, so bottom there is a stage, in that you can keep the specimen and observe it. Instead of keeping it as a handle, so the lens is fixed at the top, in this stage you can keep the specimen and observe it. So that comes under the simple microscope, 10x magnification. So any objects, so for example, if you want to see an ant, take the ant, keep it in the stage, okay, on a slide, and observe that. Observe its antenna, observe its head, observe their eyes. So like that, it is possible. That is the first one, simple. But later what they thought, so it is not enough, because he told, Antony van Leeuwenhoek itself told, 200x magnification is possible. So in that case, another lens to be used. So here the 10x, another 10x, if you are saying that, you will get 100x magnification. So here the eyepiece 10x, objective 20x, you are able to get it as 200x magnification. So in that case, how best we can increase the magnification with the help of many lenses. So in a compound, so it is called as compound microscope. So we can call it as simple compound microscope. Some of them they call it as simple compound microscope, but it is misnomer. You can call it as compound microscope with two lenses instead of a single lens. Okay, one is the eyepiece, another one is in objective. Okay, so all other parts are known to you. I don't want to explain. I will go to the next slide. Is it okay? High power objective, that is 4x, 10x, then you are going for 20x or 40x, or oil immersion objective as 100x. Okay, then clip, stage, condenser, iris, mirror, everything it is there. Okay, then what are the general principles of microscopy? How we are able to get the magnification? How we can resolve the objects? Okay, the first one is the wavelength of radiation. Okay, so in the wavelength of radiation, is it a visible wavelength, or next to that, whether it is infrared or ultraviolet or beyond that? So generally within the visible wavelength only we are making the light to pass through, refract it, and then it makes the image, the inverted image you can see, or otherwise with the two lenses you can see as such the image. This is the common microscopy principle, isn't it? So the wavelength of radiation is most important. Okay, in the electromagnetic spectrum, in the visible light, it is from 400 nanometer to 700 nanometer. So that is the color what we are calling it as VIBGR, isn't it? That VIBGR color. So this is a visible light. So UV light also used in certain special microscope, but what we discussed in the light microscope is only because light have electromagnetic spectrum. That light have, that prism has 400 nanometer to 700 nanometer. So if you pass through a prism, it is refracted, or that the pathway is deflected, and then you can converge them to a one focus and then observe the object. Okay, so this is increasing the wavelength. For example, if it is you are using the radio waves or television, so it is up to going 10 power 3 meter, whereas 10 power minus 12 also you can use up to gamma rays. So whenever you are increasing the resolving power, so you can use different kind of wavelength. So then what is the resolving power? So before that, the second point in the principles of microscopy, it is a refraction and image formation with the light. Okay, just to see that the light, you are focusing from the bottom, that light is passing through an aperture and enters inside, so it is refracted in a glass. That glass only the convex lens. Okay, so the specimen is kept, convex lens is kept in between, then there is a focal point and it enlarges the shape, size of the object and show as a reverse image. This is for single. Then if you are using another one, the same thing, it goes for a normal position. Okay, you are getting the magnification as well as you are observing the normal image of a specimen what it is kept on the stage. Okay, so the refraction and the image is another principle. The first one is wavelength. The second one is refraction and image. What is the third one? Resolution or resolving power. Okay, so it is a refractive index. Okay, it depends upon refractive index. So what is that? So it is an ability of the optical system to form the distinguishable images of the objects separated by small angular distances. Okay, so you just take your pen, make different dots in your paper. Okay, just make a dot, closer dot, then wider dots. How best your eyes are able to resolve the two different dots? If they are widely spaced, you are able to observe it clearly. The resolving power is high. But if you are very closer, you can't distinguish between the two dots. So in that case, how best you are able to resolve the distance between the two dots with the help of the particular refractive index, then that is the resolution. Okay, resolving magnification is different from resolution. Magnification, just you are increasing the size of the image, whereas here resolving power, if there are two objects close by, how best you are able to distinguish the distance between the two objects. Okay, so in the single object itself, for example, if you are observing the tail with the spermatozoa, the tail and acrosome, how they are present, and if you are able to differentiate that particular membrane, then that is the resolving power. Okay, so the resolution limit of a microscope is the shortest distance between two nearby objects when the images formed by the microscope are properly differentiated. So two closer images, how best they are differentiated, that gives the resolving power. So that has a formula. So 1.22 into wavelength divided by numerical aperture. Okay, so this numerical aperture, that hole, okay, through which only you are putting that. The resolving power depends on the aperture as well as the wavelength of the light. So by the formula itself, you know the wavelength is an important one. Okay, then the second one is the what is the size of the aperture. Okay, so this is the resolution. Okay, everybody should know what is the difference between magnification and resolution. Okay, or resolving power. Okay, what are the limits of resolution? Okay, so you start with 1 meter up to 0.01 nanometer. There are different objects are shown here in this picture, and in that you can see that with unaided eye, so you can see even from 1 millimeter very clearly, even less than 1 millimeter also sometimes you can observe it. Okay, so that is with unaided eye. Then you can go for compound light microscope between 200 nanometer to 10 millimeter. Okay, the last one you can see that compound light microscope. So it is for the 200 nanometer to 10 nanometer. Whereas scanning electron microscope 0.4 nanometer to 1 millimeter. Then TEM, transmission electron microscope, is 0.07 nanometer to 100 micron. So which one is resolution is good? Transmission or scanning? Resolution, resolving power. If you go smaller objects to be differentiated, transmission is good, isn't it? It is for nanometer to micron. Here it is nanometer to millimeter. Okay, so here the TEM is so good. Then scanning tunneling microscope, that is 0.01 nanometer to 10 nanometer. It is more precise to differentiate the two different objects. So this is that. Through this, so see that the atomic force microscopy, what we are saying, the atoms are able to be differentiated in that. Okay, then it comes to the amino acid level, or it comes to 1 nanometer diameter of the DNA, or if you want to observe the protein, it comes under the 10 nanometer. Whereas the ribosome as a organelle, if you wanted to see, so 10 to 20 nanometer you can see. Then viruses, it is around 50 nanometer scales you can see. Then typical bacteria, you can see 1 micron. Okay. Then organelles like mitochondria and chloroplast are little bit bigger, so it comes under the 5 micron or 7 microns. Then human red blood cells also coming under that, 8 or 9 micron. Then the large protozoan euglena, you can see that it is around near 1 millimeter, okay, or it may be of 500 to 600 micron. Then if you want to see a different insects, so it is more than 10 millimeter. If it is a grasshopper or locust or some other insect, it's a big in size, it may be more. Then chicken egg, so what is the diameter? Around 60 millimeter. Okay, so like that you can observe the various things. The limit of resolution varies depending upon the power of the microscope. Is it clear? The last row. Okay, so we will go to the next slide. The third important principle in microscopy is the contrast. I will say the last row, two girls sitting, one is wearing the white color and another one is wearing the red color. So it is of quite contrast. Am I right? So easily we can identify there are two persons. If there are two persons are wearing the same color, then to differentiate it is a little bit hard. Same thing here, at different thickness, at different diameter level or the closeness, we have to find out which object is differentiated from another object at what closeness. Okay, how to create the contrast. So the differences in intensity between the two objects or between the object and the background. If the background is dark, if your specimen is passing through the light, you can easily observe dark field microscopy. Okay, or if the thickness as like a face, you are create one is thin, other angle is thick, so easily you can differentiate thinner specimen with the thicker specimen. That is possible with the help of phase contrast microscope. So based on this, the resolution is determined. Besides that, if you are able to stain with different dyes, okay, so whatever you have seen, all the TS of root or stem or bacteria, everything it is stained in different colors, so that easily you can observe. You can easily give from the white background, you are observing a blue colored one or green colored one. So it is possible to contrastingly see that how contrast they are. Okay, merging. Sometimes you will use the PowerPoint slides which are merged with one another. Even I can't read from here. This is a white background with black letters. Easily everybody can see. But if you are making some other background, light bluish background with the black color, it's not possible to observe, isn't it? Same thing, contrast is important. So, use of light that is in phase increases the contrast. Different phases, the light is moving to that. So, one is easily making the object into a thick and another one thinner or different planar surface, so that you can observe in the different thickness. So, this is used for studying the living cells, phase contrast microscope. Okay, then bright field microscope. As we discussed, there are different types of microscope. Bright field microscope, it is simple, as like I told, it is a single magnifying lens. Okay, Leeuwenhoek used this to observe the microorganisms. Okay, so this is the first. Then second one is compound, series of lenses for magnification. How many lenses now we are using? In a compound microscope, what you are observing in a class? Two, isn't it? Two. Okay. So light passes through the specimen into the objective lens. Then you can increase the power resolution also using the oil immersion. What is the principle? We will see later. Then it has one or two ocular lenses. Okay. Total magnification equal to magnification of objective lens into magnification of ocular lens. Okay, 10 into 10, 100, 10 into 20, 200, 10 into 40, 400, 10 into 100, 1000. That is the maximum possible in the compound microscope. Most have the condenser lens. Why need a condenser lens? To avoid the dust, to avoid the thing, and also focus the light to move through a particular orifice. Okay, so for that direct light through the specimen. Okay, this is the way how the image is produced. So compound microscope are easily portable, affordable and easy to use, and it goes up to the magnification of 1000x. Okay, and the light source comes from the bottom and passes through the microscopic lenses and makes fully visible. Okay, it is view the objects at the cellular level also, it is possible. This is the one what you are using. Okay, next. So this is the whole parts, already you have seen, fine adjusting knob, base, then coarse focusing knob, illuminator, diaphragm, condenser or stage, objective lens, then arm, body, ocular lens or eyepiece, everything it is given here. Is it clear? Okay, so here based on the number of ocular lens, we enter monocular microscope, binocular microscope, trinocular microscope. Okay, what is the need for trinocular microscope? With the two eyes we are observing in binocular. What is the need for trinocular? So in the another one, they will attach the camera. Okay, they can take photographs, or they can attach directly to a computer. So in the computer you can see the digital images of that what you are observing in the slides. Okay, so next. So what we are saying, in the higher end resolution, the 1000x, we are saying that the oil should be kept, cedarwood oil or some oil should be kept for 100x. Okay, so how? If there is no oil, the refracted light rays lost to the lens, whereas when you keep the oil, more light enters the lens. This is the principle. Okay, you can see that the refraction on the both the sides doesn't occur because of the oil you are putting a drop. Immersion oil, here in this layer you can see an immersion oil. So more light enters the lens, whereas here without the oil, glass with the cover slip, so here the refractive light is deflected on the sideways, so it is not focused towards the lens. Okay, then you can see it. This is the principle for oil immersion. Okay, then what is the stereoscopic microscope? Okay, it has a light source on the top to illuminate the specimen. Light source, generally what we are having at the bottom, we are putting the small lens, isn't it, that receive the light, okay, or you make the different modes of making a light source. So here, to illuminate the specimen, the stereoscopic microscope have the light source at the top, causing the reflection on the microscope lens. So here the maximum magnification is up to 50x. So the light passes through the microscope tubes, create the image which provides three-dimensional image also, and improvement of the flat image which is found in the compound microscope. Okay. So where it is used, everything it is generally what stage is at the top. Am I right? In our microscope, common compound microscope. Here the stage is at the bottom, all the adjustments are at the top. Okay. So for dissection, so you can keep it in a Petri plate and dissect the eyes or any of the things for insects, or if you are dissecting a particular worm or dissecting a bacteria, so you can easily take a pollen of a flower, dissect it and observe under the microscope. So it is used for dissection, even for the coin appraisal or gem mineral study. In the geology, the microscopes are used in plenty to study the structure of that minerals, okay, and also in entomology, study about the insects, okay. And so generally, if you go to a shop where their wristwatches are getting repaired, okay, what they are doing, first they will use a single lens keeping on their eyes and see that, and if it is not possible, then they have the microscope like that and keep the entire circuit in the microscope and see that with a small pin, rotate it and observe the different parts where the defects are present. Okay. So the stereoscopic microscope, you can see that eyepiece lens and the objective lens and the light source. So the unrefracted or hollow cone of light is passed. Okay. Then dark field microscopy. If it is object is pale, so if the background is dark, you can easily the contrast possible. Okay. So the light rays are scattered by the specimen, entered the objective lens. Generally, what is the normal one? Only scattered light entering the objective lens, whereas in the normal one, main light through the aperture it is going and focus on the object. Okay, the specimen appears light against the dark background, that is the dark field microscopy. So it increases the contrast and enable the observations for more details. So this is under the dark field microscopy. So just observe in the dark field how they are creating the dark field. In the center it is dark, they are putting some dark field stop. That is in the center you can observe that it is a dark, complete dark. So only through this the light is focused. So what will happen? So this is the field and this dark field, over that the pale specimen are kept and you can easily observe them. Okay. Then phase contrast microscopy. Okay. Already we discussed, here the phase plate is used. Okay, and the light rays in the phase produces the brighter image, while light rays out of phase produce darker image. See? You are out of frame. Why we are calling it as? If you are frame, photo frame, or when you are taking a photograph, so if you are not in a main frame, so you are out. Similarly here, so you are making that outer one with the darker and the inner one with the lighter, based on the adjustment with the light, how they are entering in the diagram you can easily see. So this is the inverted phase contrast microscope. Why we are calling it as inverted microscope first? Why we are calling this as inverted microscope? Yes, stage is in. Where is stage? Here you can see stage. Okay, so the eyepiece are below the stage, isn't it? Even though it is here, that is below the stage only the tube-like apparatus. The eyepiece. Generally, eyepiece above the stage we will see. Okay, so this is in the dissection microscope for easily keeping that, and the condenser, phase ring, everything will be below, but here it is above. Okay, so the inverted microscope, and in that you can see a face plate. To show the face plate only they are keeping like this. This is a face plate, you can adjust. So there is a ring like structure, and through the rings the light are passed, and it may be into different kind, so that it makes the contrast possible. Okay, so it is called, with the help of the face plate they are making the different images of different thickness. Okay, so then there is another one with the same concept, contrast concept itself. So it is called differential interference contrast microscope. So this is not much used in the cell biology, even though it is there for various type of other one. So here the fluorescence microscope, everything is used, and here they have the two different spectrums. Okay, modified Wolfson prism. Generally the prism principle is used, what we are seeing as a small one, but here the two beams are traveling in two different directions. Are you able to see that? As like a DNA helix, two beams are going in two different directions, and then it is converged in the light to the eyepiece, then you can see the specimen. So okay, this is one of the microscope available, that is enough. Then what is the principles of phase microscopy? Okay, how the rays are able to present in the phase, light rays, then how they are out of phase. Are you able to see first wavelength and the second waves? The waves of both rays in phase, rays out of phase. What you are seeing the observation? ரெண்டும் ஒரே மாதிரி தான் சார் இருக்குன்றீங்களா? Just to see that closer you can see and tell. Rays out of phase, upper, rays in phase, the crust. The crust is important. Just to see that the crust is more converged, and then they are able to give the good image, the light passing. Okay, and just ray deviated by the specimen 1/4 of the wavelength of out of phase, and deviated here it is half wavelength out of phase. Okay, so 1/4. So in that case it will give you a closer picture. Okay, and then depending upon this you can make different contrast, different thicknesses. You can see a cell wall or epidermal layer, you can also see the vascular bundle, or if you are taking a cell you can see a plasma membrane, at the same time you can see a nucleus under different contrast. Okay, so this is the basis for contrast, phase contrast microscope. Then fluorescent microscope. Okay, so by the name itself it is self-explanatory. What is fluorescence? What is fluorescence? I think ladies should know. Fluorescent color you are selecting for your dress, isn't it? What is that? Or the painters, gents, fluorescent painting. Fluorescent colors are used for painting. They glow at the dark. Am I right? They glow at the dark. Okay, so here they are go and bind to a particular organelle, and easily it can be differentiated from other. Okay, for example Janus green is added, so it is added to the, it go and specifically bind to the mitochondria, so that easily differentiated from other organelles. And when the light is very less, even then you can easily observe the fluorescent colors. Okay, some of them naturally fluorescent. Can you tell any one naturally fluorescent? It emits light naturally. Yes, yes, somebody is telling. Huh? Phytoplasma? Phytoplankton. Okay. Have you observed the firefly? Minmini puchi. Okay. At the abdomen, they emit light. Okay. That is light. But fluorescence also comes from certain specimen. That is, they are giving a different colors when the light enters in that. Okay. Bright colors. So, this is used for staining. Okay, and also immunofluorescence used for pathogens. That is specific fluorescent material tagged with certain material. That is, you know, the key and lock hypothesis. Enzyme and key and lock hypothesis for what? Enzyme and substrate. Isn't it? You are very good in chemistry, am I right? Enzyme key and lock hypothesis. Okay, enzyme substrate complex. Similarly, antibody antigens. Okay, so in that, if you tag one with the particular fluorescent color, you can see a fluorescent images. All other fields are not visible, but your chromosomes alone are able to seen if you are doing with the fluorescent staining. Okay, so like that it is possible. Okay, so 4 kinds of light microscopy in that: bright field, dark field, phase contrast, as well as the Microscopy. You can see the variations between that. Okay, then immunofluorescence also. That is, here the fluorescent dye is added to the antibody. Then wherever the antigen is present, the antibody go and bind. So what happens after binding? That part is give the fluorescence. Easily you can detect where the antigen is particularly present. Is it clear? Just observe the red color and green color. Green color are the fluorescent bodies. The red color is antibodies. So they carry the fluorescent dye, and when the cell is present, wherever that particular antigen is present, this antibody go and bind, and you can easily observe under the microscope. Okay. So right, this is immunofluorescence. Okay. Then confocal microscope. In the light microscopy itself, confocal microscope. So here also the fluorescent dyes are used, but the resolution is increased using the UV lasers, because the emitted light passes through the very minute pinhole aperture, so that the image is visualized or constructed as a 3D image, and then digitized images you can get it in case of confocal microscope. Just we will see how. So here the lasers are used to scan the specimen, create a high resolution, high magnification. Both are high. Sometimes the magnification may be high, the resolution may not be high, but in this case both resolution as well as magnification is high. Okay, and so the selection by scanning the specimen and creating the section, so that you can build the 3D image and without any physical destruction. And here you can observe the living cells, okay, or embryos marked with some dyes, fluorescent dyes. Okay, here they can reach up to the magnification of 2000x. Up to that, all the optical microscopy, what is the x magnification possible? 1000x. So here improvement, okay, 2000x. So this is the diagram how the detector is taking the digitized image. So you can see a first focal plane where the specimen is kept, and through the objective lens, here the laser is treated. Then because of the, and then it the laser goes through the pinhole and there is a mirror, and with that they are able to make an image, and with the confocal pinhole, so it is connected to the detector has the digitized image. So this is in case of confocal. Okay, what is the electron microscopy? Okay, so what is time? Is it? We'll finish it. Huh? 20. Another 10 minutes. We'll finish. Okay. So electron microscopy. So here everybody know the accelerated electrons are used instead of light energy. Okay, and through which they make the digital image. The magnification limit is so good, that is 10,000 x to 1 lakh x. Okay, and we can view bacteria, virus, internal structures, cell structures, molecules, larger atoms. So everything it is possible, and resolving power also closer than 200 nanometer. If there are just placed 200 nanometer distance, you can easily distinguishable. Okay, and but only one disadvantage is, since because you are passing the electron, that may kill the cells. You can't see the live cells. Always you have to make the fixatives, everything, and prepare the specimens. After that only in a slide form you can observe under the transmission electron microscope. Okay, so then there are two: transmission electron microscope and the scanning electron microscope. Both you are going to see in the last practical class we have in nanotechnology department. Both the microscope are available: transmission electron microscope as well as scanning electron microscope. What is the transmission? In the electron microscope, what is the principle? What is the difference between light microscopy and the electron microscopy? Okay, so just if you take that, in the electron microscopy, the electron gun is present, that is the source, and then the condenser lens, then condenser aperture, through that the rays coming, then it hit the sample, then they have the objective lens. So with the help of the objective aperture, so the immediate lens and the projective lens makes the image to occurring in the fluorescent screen. So this is the main part of electron microscopy. So in that, the main, instead of light, we are using the electron. Okay, so that easy high resolution as well as the magnification. So in the case of the TEM, it passes the electron through a thin specimen, and the light pass through the specimen. The electrons pass back and forth through the microscope's vacuum chamber, so that only it is bigger in size. So this is like a tabletop recently come, but earlier if you observe that entire room is allotted for transmission electron microscope. Very big beam of electrons will come, and in that they are completely covered with a vacuum chamber. Okay, and it is stronger than the same microscope, up to 1 nanometer resolution and 500,000 x. Okay, so just observe it. It looks like a rocket launching pad. Okay, so it is just to observe the specimens at a very good scale, final image in a fluorescent screen, how best you can observe that particular object. For most of the viruses, they are using the transmission electron microscope to observe the various characters, and also it also gives what is the composition of that particular in the head capsule, the DNA, RNA, or the proteins, what are they. So everything it can be attached to another apparatus, and then see that how best you can see that. Then scanning electron microscope, the image is scanned. Okay, and the specimen is kept on a stage in a vacuum chamber, which removes the electron inhibiting air to aid in the acceleration. Okay, and this makes up to 10 nanometers resolution and magnification of 30,000 x, whereas the TEM 5 lakhs or 500,000. So this is only 30,000x. But on the surface, you are not processing anything. You are just taking, even you are just to take a hair and keep it under the surface in a slide, and even up to keratin pigment you can observe up to 30,000x. Okay, so the cells are present in your hair at the bottom. It is a very small cells. Okay, so that only the hair is used as one of the evidence for crime cases. How they identify a person with just a small removed hair? At the bottom there is a cell, so you can extract the DNA from that and identify a person based on their DNA. Okay, so you go and plug your hair and see the cell whether you are having that, and okay, so that is the most important, even for dogs or whatever. Okay, so it is a, so this is sample. You can observe that electron gun is coming and it forms a beam, and through the condenser lens it go and scan the coil, and on the objective lens the magnifications, everything is controlled, and the scans are generated and they are displayed, amplified, and then give it as a bigger image. Okay, same image. Just observe that it is a very small organism, but you can see that how brilliant they are able to take the various stages of the fungus, aspergillus, or the paramecium, or streptococcus bacteria. So everything, the same images. It is on the surface, but on the inner details you wanted to see that, you have to go for TEM. Okay, right. Then the last one is probe microscopy. Okay, it magnifies more than 100 million times. Okay, 100 million times. So in that there are two important one: advancement scanning tunneling microscope or atomic force microscope. The scanning probe microscope or tunneling microscope, less than 1 nanometer resolution, you are able to keep it, as like here also the probe is attached and it scans to a particular specimen, and it measures via the laser and send the information to photodiode and then convert it into digital image. This is also used in nanoscale, and the cells and molecules are studied with the help of this microscope, scanning probe microscope. Okay, so just you can see a DNA as well as the enzyme, how best they are able to take a photograph with the help of a scanning probe microscope. So how you are saying it is a DNA, it is a protein or the enzyme? The probe is attached. A particular part go and bind specific to DNA, specific to protein, so that can be imaged. Okay, so the probes are different types, so don't bother about that. It may depend upon your molecule. You can use the different kind of probes and then attach to that. The colored probes, okay, with the fluorescent colors, everything it is there with a short sequence. There is a, in the DNA means a probe is a complementary DNA. A always pair with T, C always pair with G. So like that you take a fragment, you attach that fragment with a fluorescent tag or a probe. Okay, then wherever the complementary sequence is there, go and bind in the specimen. So, and that shows a double helix, double stranded DNA. So that can be realized with this specimen. Okay. So there is an last one, X-ray microscopy. So that uses the electromagnetic radiation in the X-ray, and X-ray is allowed on the sample, and so the magnification is done. There is a lens which was recording in the case of CCD camera. Okay, so this is the principle. Through the X-ray they are passing, so it's called X-ray microscope, exposes the film. Okay, X-ray, how they are taking, they are allowing your X-rays to see, scan the particular part. So if this part is to be scanned, so immediately they will ask you to keep like this, and the X-rays passes through that, and on the other side the image will be done, and that is taken in a what, black colored sheet. It is in a normal one. So here what they are doing, they are recording in a CCD camera that object image how it is. So they record in that, and then they will give it as a digital image. Okay, so it is possible. This is the X-ray microscopy. As I told, the last one is atomic force microscopy. Just you know the name, atomic force microscopy is used to study the atoms which are present in the different molecules. Okay, and in that the reflected laser beam are able to give the various type of cantilever, and which are acting as a probe, and with the probe you can scan the sample and you will get a bigger picture like this. So this is the highest advancement now, atomic force microscopy. But is it possible to take our finger and then observe under a atomic force microscopy? It is not possible. The finer, the very small, very thin objects, so you have to make a preparation and then observe some 1 lakh times magnification. So if you magnify 1 lakh times our finger, it looks like the higher it is a high resolution image than the elephant's leg. Am I right? 1 lakh times higher image. Similarly, you have to see that what is the relative size, and also always when you are drawing the diagram, or when you take a photograph, or when you are scanning some of the specimens, you have to make it as how many x magnification. Okay, how much resolution you are able to take it. Okay, now it is in pixels in camera. Okay, how much pixels you have used and what type of camera used. Same thing in the microscope, you have to write it how much magnification you have observed, because if you are comparing two different objects and two different magnification, if you are saying this is bigger and this is smaller, you are committing a mistake. Okay, under the same magnification how it looks like. So that is the most important one. Okay.